Good morning, students. This is our chapter 11, Biotechnology, its principles and processes. Today we'll study about preparation and insertion of recombinant DNA into host cell and polymerase chain reaction. First of all, we'll see what is host cell. The cell to which desired new gene is introduced is called host cell. Any living cell can be used as host cell, but mostly Escherichia coli bacterial cell is used as host cell in genetic engineering because it's simple prokaryotic cell, it's non-pathogenic bacteria, and it can be cultured easily in the lab. It has very short lifespan and it contains self-replicating plasmid. And the plasmid of Escherichia coli can be easily handled as a vector. The steps involved in recombinant DNA technology are: first is isolation of desired DNA, and then fragmentation of DNA by restriction endonucleases. Example is EcoR1. Isolation of a desired DNA fragment, then ligation of DNA fragment into vector with the help of DNA ligase, and then transferring the recombinant DNA into the host, culturing the host cell, and extraction of desired product with the help of bioreactors. In this slide, there are three steps. In first step, you can see plasmid. Plasmid you all know, and it's having a cleavage site. Then it's donor DNA, and both the plasmid and donor DNA are treated with the same enzyme. Just like here you can see, EcoR1 is used as endonuclease. So a cut will apply on the palindromic sequences. So the next is hybridization, means plasmid and this donor DNA, the fragment taken from donor DNA are hybridized, and then DNA ligase is used. Then this is going to form recombinant plasmid. This is one more slide showing cloning into a plasmid. You can see plasmid and a foreign DNA on which both EcoR1 is being acted. Endonuclease is restriction, endonuclease is acting. Now hybridization and then with the help of DNA ligase, then recombinant DNA is formed, and this recombinant DNA then is introduced into the bacterial cell, and which is already having bacterial chromosome, and a new plasmid is introduced in it. And now cloning is done, and these are bacteria plated on medium plus antibiotic. Only bacteria containing recombinant DNA will grow, and these are formed in the lab or in the bioreactors, and a product is formed like this. And likewise we can form insulin etc. in the lab. This is working mechanism of polymerase chain reaction. A single polymerase chain reaction amplification cycle involves three basic steps. First is denaturation, then annealing and extension. Denaturation is basically the target DNA is heated up to high temperature, that's up to 94 degree to 96 degree Celsius, resulting in the separation of two strands. Each single strand of target DNA then act as a template for DNA synthesis. The next step is annealing. Annealing means to join. In this step, the two oligonucleotide primers anneal to each of the single-stranded template DNA. Since the sequence of primer is complementary to the third end of the template DNA, this step is carried out at a lower temperature, usually 40 to 60 degrees Celsius, depending on the length and sequence of the primers. And the third step is extension, where we'll take DNA polymerase synthesizes the DNA region between the primers using deoxynucleotide triphosphate and magnesium ions. It means the primers are extended towards each other so that the DNA segment lying between the two primers is complete. The optimum temperature for this polymerization step is 72 degrees Celsius. So what are the applications of PCR? First is detection of pathogens. PCR is being used in the detection of HIV, virus of AIDS, etc., diagnosis of specific mutations. It's used in DNA fingerprinting, and it's used in prenatal diagnosis. It's useful to detect genetic diseases in fetus before birth, and it's used in paleontology. PCR is used to clone the DNA fragments from the mummified remains of humans and extinct animals like woolly mammoth and dinosaurs etc. इसी तरह से they are used in gene therapy. PCR proves to be of immense help in monitoring a gene in gene therapy experiments.